Hello everyone. Thank you so much for this opportunity to present at MVIF. I'm Vedanth, and today I'll be presenting a Python tool called argNorm for the normalization of antibiotic resistance gene annotations to the Antibiotic Resistance Ontology. Before I get started, I'd like to acknowledge all my amazing co-authors who you can see on screen, and I'd also like to point out that a manuscript detailing argNorm has recently been published as a preprint, which you can read by scanning this QR code down here. A challenge that we currently face when studying antibiotic resistance in genomes and metagenomes is that ARG databases use inconsistent gene nomenclature. This makes it hard to compare results from different ARG annotation tools and hinders effective analysis and collaboration in antimicrobial resistance research. This inconsistent gene nomenclature is really of two types. The first one is when ARG databases assign different names to the same gene. For example, this particular gene conferring resistance to aminoglycosides has 6 different names across 6 databases, meaning a naive string comparison between them would be incredibly challenging. The second type of inconsistent gene nomenclature is when ARG databases assign the same name for different genes. For example, the DeepARG and CARD databases both have a gene called ERMA that confers resistance to erythromycin. However, their sequences are very different, with just 17% identity overall. In fact, this discrepancy can be traced all the way back to 1985, when 2 independent research groups reported 2 different genes conferring resistance to erythromycin. However, both of these groups ended up calling their respective genes ERMA. And so this naming conflict has made its way into our current ARG databases. Our software, argNorm, solves this issue of inconsistent gene nomenclature by mapping antibiotic resistance genes to the Antibiotic Resistance Ontology created by the CARD team. The Antibiotic Resistance Ontology, or the ARO, is a controlled vocabulary of formalized relationships between antibiotic resistance genes and other resistance determinants. The ARO provides unique identifiers called ARO terms to specific antibiotic resistance genes. And by mapping antibiotic resistance genes to these ARO terms, argNorm provides consistent and comparable ARG annotation. argNorm supports all antibiotic resistance genes in our 8 databases that we support. Furthermore, by leveraging the resistance information available in the ARO, argNorm also provides drug categorization for antibiotic resistance genes. Moreover, by using other Python libraries such as Pronto with argNorm's ARO mapping, users can further explore resistance information in the ARO. argNorm is a completely open source tool. We've created a Python package for argNorm. We have a bioconda recipe for easy integration in conda environments. We've created a Nextflow module for argNorm for easy integration in bioinformatics workflows. And argNorm is also part of the FuncScan pipeline to analyze genomes and metagenomes. We've created a YouTube tutorial for argNorm to help users as they use the software. And if you have any questions, please feel free to reach out through email and connect via LinkedIn. And with that, I'd like to conclude my presentation. I'd love to take any questions during the Q&A session. And thank you once again for this amazing opportunity to present at MVIF.